In this video, we're going to continue to look at mechanisms of DNA repair. And we're going to start with mismatch repair, or MMR. So mismatch repair is when you have mismatches, insertions, deletions of up to 4 nucleotides. As we can see here, after DNA replication, we have a couple nucleotides in a row that are not matching correctly, and we have this kink in the DNA structure. So we're going to see how this works in E. coli because it's a lot simpler than eukaryotes. So E. coli, there's 3 different proteins. The first one is mut-S. So mut-S will come and bind to the mismatch bases, and there will be 2 copies of mut-S that binds. Once mut-S is bound to the DNA, mut-L will come and bind to them. So now you have a mut-S2, mut-L2 complex. And what they're going to do is that they're going to go along the DNA in both directions until they find a GATC palindrome. So they'll just search the DNA until they find this palindrome. Once they find the palindrome, a mut-H will come and it will be recruited. And what mut-H does is an endonuclease, so it will find the DNA strand that's unmethylated and make a nick on the 5' side of that DNA. And the reason why it chooses the unmethylated, because that is the daughter strand, right? So on the GATC palindrome, as you can see in this image, blue here is the parent, red is the parent, blue is the daughter. And we have a methylated GATC, which means that if there is a mismatch, it's due to the daughter, because this parent DNA has been around. This is serving as our template for DNA synthesis. So we accidentally added in incorrect base when we were trying to copy our parent. So please come and cut that away, cut the unmethylated DNA. So that gets chopped off. And once we have that chopped off, look, we have a 3' hydroxyl. Now we can have DNA pol 3 come in and try to recopy this parent DNA and then seal it via ligase. So sometimes these errors in our mechanisms to try and fix DNA errors will actually cause new errors. So let's look at Pol-Eta. Pol-Eta can work through these various, some of these lesions. For example, if you have a thymine-thymine dimer that's not repaired, Pol-Eta, what it will do, it will just find this thymine-thymine dimer and put 2 A's across from it. We call these translesion polymerases. They'll work across these lesions. Now, these translesion polymerases are relatively inaccurate because they don't have any proofreading, and since they have no proofreading, they have an error 1 every 30 times they try to put into a base. However, the reason why we have such crappy polymerases is that it's better than not copying your DNA. Having no DNA copy is way worse than a 1 in 30 chance of putting in the wrong base, because not copying your DNA is not a successful, you can't successfully split your cells, so that's dead cell there. And these translation DNAs, we usually have more copies than your normal polymerases. In fact, humans have 10 different copies of these polymerases that can work past these lesions. Now, if you do have an error, as we just saw in the previous slide, the mismatch repair system could fix it. And these are not that processive, so they only work on a few base pairs and then fall off. So hopefully it won't be widespread errors. And one of the reasons why these aren't so accurate is because they have to work past DNA that has weird geometry because of these lesions. So their active site is much more open. So if you remember when we talked about the polymerase in E. coli, the reason why we made a correct base is that we were making correct hydrogen bonds between the nucleotides in the enzyme. Here, we don't have that because our active site is much more open, so we can't have that check to see that the right bases are being added. Alright, so let's talk about a little more DNA damage now. So end joining, or how to fix double-stranded breaks. So double-stranded break can be quite bad, but 5 to 10% of cells that are divided in a culture have at least one chromosome break at any given time. So it's not that rare, but it is cell death if it's not fixed. And there's two general mechanisms that cells use to fix a double-stranded break. One, recombination repair. Two, non-homologous end joining, or often called NHEJ. So I'm just going to go through the very, very basics of NHEJ here. Chapter 26 goes into that a lot more in depth, but this is basically the 1,000-mile view of what happens. So your DNA breaks, both strands break. What happens is that you take these broken DNAs and you try to line up the ends. Anything that's like overlapping or frayed, you're going to excess trim that off or fill it in. Then you're going to ligate. So it's just a way like, oh, my DNA broke. I'm going to try and just rearrange the ends again, fill in what's missing, and hopefully I didn't lose any bases during that. That's the general basis, and for right now, just knowing that NHEJ is for double-stranded breaks is good enough. Like I said, there's a much more detailed mechanism for that later on. But the core of NHEJ is this Ku protein. This is that protein with DNA. This DNA is in the black and the light blue there. And what happens is that this protein is just cradling our DNA inside of itself, but it's not really touching any of the bases. It's just fitting the major and minor groove. So it's coming along, and if you come to a break, this protein can recognize that because it's just cradling the DNA, and it will go and start the NHEJ schematic. And this protein, like I said, will line the DNA where it breaks, and the ends can be accessible to nucleases, polymerases, ligases, all we need to fix that break. And we'll go into this mechanism in much more detail later. Last thing we're going to talk about in this video is the SOS response. So sometimes damage can be so great that you have a whole nother system to try and alleviate cell death. So in E. coli, this is called the SOS response. So if the SOS response is active, you will stop all cell divisions, and you're going to put all resources towards repairing your damaged DNA. The proteins that are big in this are LEX-A, which is actually a repressor of the SOS response, and REC-A, which is DNA binding and regulates the activity of the SOS response. So normally LEX-A is on the SOS gene, so the SOS genes cannot be expressed. So LEX-A is making sure that our genes are not being expressed. However, if you have DNA that is damaged and unrepaired, eventually REC-A will come and bind to that damaged single-stranded DNA. Once REC-A is bound to the single-stranded DNA, this complex will then go and interact with LEX-A. LEX-A will then cleave itself, and it will become inactive. That is, it will move away from the SOS genes. It's no longer bound to them. And since there's nothing inhibiting the SOS genes to be expressed now, these SOS genes will be expressed. And these SOS genes are REC-A, LEX-A, these nucleotide excision repair genes, UVRA and UVRB. And you also get new polymerases that you're going to make, PAL4 and PAL5. And these are translation polymerases that do not need a template. They will just start adding bases to your DNA. So these are super error prone because you don't know what you're matching to. You're just randomly throwing in nucleotides. But if you're in the SOS response and these polymerases are only activated after 50 minutes of this SOS response being active, that means you have DNA damage, cannot be repaired through any of the other methods we talked about. So your last resort, throw nucleotides in there, and you're going to have to live with the mutation, because your loss of function of a protein is much better than certain death. Sure, you might die from doing this, but you were going to die regardless. So might as well take that chance and just start throwing nucleotides in there to at least get a functioning genome back again. And what happens is that under these extreme conditions, your rate of mutation skyrockets because you have these new polymerases just adding in nucleotides. Well, what could happen is through one of these random mutations, you get a new skill or an enzyme that can do something different. You get some adaptation to these new conditions. Now you can live in these conditions. So, evolution. And that's happening because you were on the brink of death and had these new polymerases. Alright, and with that, that's the end of this video. I will see you in the next video.